madam, please. Yes.
Are you able to see my screen, all of you?
Yes, ma'am.
Okay, good.
Madam, you can start.
Yeah.
Thank you. Thank you. Okay.
So today I'm going to discuss one of the important topic and a very interesting topic, forthcoming, developing topic in the world. This has taken shape. All of you are aware about the Human Genome Project that was completed in the year 2003. And really the post-genomic era led to so many developments. We were able to think in a totally new perspective, and application of the human genome data for clinical practice and biomedical research led to a great paradigm shift. And the mutations that could be identified by the Jiva studies or by the other large-scale sequencing techniques that are available now, what we are going to call it as the third generation sequencing technology that we are today we are witnessing, and we are utilizing it. And also the cost of the genome sequencing. For example, if you are going to see here the protocol or the amount that was spent to do the sequencing of Human Genome Project, nearly $3 billion, and the estimated time was somewhere around 13 years. So with the third generation sequencing technology, in other words, we are going to call it as the NGS sequencing technology, the cost of the genome sequencing has come down approximately to 25, anywhere between 25 to 35,000. And also the time that is required to sequence and completely annotate a human genome is less than 3 days. So this is what is the post-genomic development that has happened after the availability and the successful completion of the Human Genome Project. So here you will be able to see here two prestigious scientific journals, Nature and Science. So generating the first sequence of the human genome they published, and then provided fundamental information about the human blueprint, and then which has since accelerated the study of human biology and improved the practice of medicine. So this led to a lot of new dimensions and new school of thought were generated. So Human Genome Project is one of the greatest scientific feats in history that we have witnessed. And this project was a voyage of the biological discovery led by an international group of researchers looking to comprehensively study the DNA of a genome of select set of organisms. It was launched by October 1990 and completed by the year April 2003. So all of us, this information, all of us are well aware. So what is the development that happened after the Human Genome Project is a very remarkable development that we are witnessing. So for example, the HapMap database, formation of the HapMap database, 1000 Genome Project, and the ENCODE genome database. So these are some of the remarkable developments that we have witnessed. So with the completion of the reference human genome sequence, the whole attention was, say specifically the perspective of thinking was focused towards the cataloging of the mutations, or in other words, what we call as variations in the sequence, and correlating these variations into disease. So this was the direction, especially as you are aware, with respect to cancer research and with respect to the Alzheimer's or Parkinson or the various other diseases. Now the entire scientific fraternity who are working with the human molecular data, they are focusing on identification of novel variations in the gene and relating that and deciphering what is the change that is happening in the metabolic pathway, and specifically what is the difference between a healthy metabolic pathway and that of a disease metabolic pathway, and how exactly the disease is being expressed. So that is why the human genome variations we can very broadly classified into two types, that is the insertion, deletion, and duplication. And on the other side, you're going to see the single nucleotide modifications that we are going to observe in the genome. That is very critically, it is called as SNP, single nucleotide polymorphisms. And another major variation, if we are going to come towards the insertion, deletion, and duplication process, so that we will be able to say again classify depending upon the base pair. If the insertion or deletion or duplication is somewhere around, say, approximately around 10 base pairs is going to happen, that you are going to call it as the short tandem variations or STRs. Then if it is specifically higher than 10 base pairs, between specifically 10 to 10 BP base pairs to specifically less than 1000 base pairs, if we are going to see specifically between 10 BP and 10 KB, if we are going to see over here, we are going to classify it under the VNTR sign and the line mutations, we are going to call it. And the very long mutations ranging between 1 KB to 1 MB, they are called as specifically the CNVs, copy number variations. So this is how the variations in the human genome are very critically classified. And the very crucial process that has to occur is the very accurate analysis of the genome data that we have to very specifically generate, and then analysis of the data, and then gene expression data, what are the proteins that are expressed over here, and the proteomics and metabolomics data is going to come over here. And then the environment and the lifestyle and the food habits. So all of them, they are going to play a very crucial role. So broadly, I have divided my slide into two components, if you are able to see. One is completely a computational analysis part that's called as the bioinformatics. And another one in clinical practice, specifically the diagnosis of the disease and the collection of the data and the population that is affected by that particular disease. All this information and statistics are taken. So this is specifically comes under the health informatics. So the health informatics and the molecular biology and the computational analysis and genetics put together, you are going to call it as the bioinformatics. So the analysis of the patient data for the specific variations, in other words, is the need of the hour. So what exactly, how are you going to utilize this genomic data and the specific variations? For this, a new word is being formed in the field of clinical medical practice. It is called as the precision medicine, a form of medicine that uses information about a person's own genes or proteins to prevent, diagnose, and treat a disease. So specifically, this is being practiced in the treatment of the cancer. So generally, one treatment, one line of treatment, or one line of protocol is not going to fit all the patients. So especially in cancer, what exactly is the problem, and what is the type of cancer, and how it is pronounced in the patient's body. So by what are the genes that are expressing in the cancer tumor. So this information can be collected, and thereby precision medicine can be line of treatment can be formulated. So in cancer, precision medicine uses specific information about person's tumor to help to make a diagnosis, plan a line of treatment, find out how well treatment is working to make the prognosis. So this is being practiced, especially with respect to the cancer treatment. So here, whatever I said symbolically in the form of a infotainment I have given over here. So specifically, you will be able to see here the cross section of the population you are going to see over here. And then the patients are grouped by disease, subtype, demographic features, and then clinical features, and then the biomarkers each one of them are carrying. So based on that, you have to classify them. So after that, the personalization to what line of treatment they are going to fit in by seeing their behavior, food habits, and other living style, lifestyle. So all these properties can be taken and they will be grouped, and a specific line of treatment can be given. So here, what is the principle of personalized or precision or whatever the targeted medicine you are going to call here? The same, the patient group will be there, and the diagnosis is more or less the same for that particular disease. But when we are going to give, we'll be able to classify them into more or less 4 or 5 groups. So when we are going to give a line of treatment to a certain group of people, what is going to happen? Some group may show that the drug toxic side effects they may show, and but still the line of treatment will be working in them and it will be controlling the disease. So this is one group. But on the other hand, you are going to see one more group over here, that the drug is not toxic, neither it is beneficial to them. So for example, let me say that this is a cross section of people who are diagnosed with a particular type of cancer. So when you are going to give, let us say, a X drug, if you are going to say treat them with, what is going to happen? That X drug might be toxic to a group of people, but still it will be controlling the disease. But whereas the other few patients, neither it is toxic in them, nor it is beneficial, nor it is controlling their problem. So this is another few people are going to show such a type of response to a particular line of treatment. And one more group is there. The drug is toxic, but it is not at all beneficial. It is creating adverse drug reactions in the patient's body, nor it is controlling the disease. But on the other hand, you are going to see a fourth group over here, same diagnosis, same line of treatment. The fourth group, they are going to show the drug is not at all toxic in them. They are not showing any adverse drug reaction, and the drug will be highly beneficial, and their disease is being controlled. So these are the 4 type of events, 4 type of reactions that we are going to observe when we give a line of treatments to the patient who are diagnosed with the same disease, and the same line of treatment is given. So then how are you going to handle? The drug is toxic but not beneficial. The drug is neither toxic nor beneficial. Yes, this line of treatment is going to suit this group perfectly. But the drug is not toxic and not at all beneficial. But on the other hand, the drug is showing ADR reactions, but it is not at all beneficial. So there are these 3 groups that has to be addressed, and they have to be decided with a different line of treatment. So precision medicine is a emerging approach for disease treatment and prevention that takes into account individual mutations in the genes or individual variation in the genes And also the environmental factors has to be taken into consideration, and lifestyle for each person has to be taken into account. And this approach is fairly where you are going to see very minimum adverse drug reactions we are going to see. This approach will allow doctors and researchers to predict more accurately which treatment and prevention strategies for a particular disease will work, and which group of people, what type of line of treatment has to be given to them. So we will not be able to, for example, if we go to a ready-made shop, what is going to happen? Uh, there will be, say, let us say, if we want to buy a T-shirt, there will be a L size will be there, S size will be there, M size will be there, XL will be there, XXL will be there. So when we are going to try these T-shirts, what is going to happen? If everything is all right, the length of the T-shirt will not be correct. If the length is correct, some other component will not be correct. So that is why we cannot very particularly say that if we apply the same logic over here, we cannot say that one size fits each and every one. So one line of treatment, one line of treatment protocol is not common to each and every patient. So seeing the reaction or seeing the mutations in the gene and what exactly is the food habit, environment, lifestyle, all these things has to be taken into consideration, and accordingly the disease treatment and prevention strategies has to be developed. So for the average person with less consideration for the differences between individuals, so this is what the drug companies and even the uh, drug federation approval body has also approved. So drug discovery, design and trial pipeline, if you are going to consider and study carefully for certain time, so specifically a pharma company is going to come out with a decision to develop a new drug for a particular disease, and they are going to scale down upon the novel candidate drug molecules, and then they are going to do the cell assay development, and then test and identify the compounds about their QSAR activity, ADME parameters will be taken into account over there. And then it goes for the animal model testing for efficacy and toxicity, cost evaluation for large scale production, and then human phase 1 trial. The human phase 1 trial may go up to fourth phase also it goes now. So phase 1 trial, phase 2 trial, and the phase 3 and the phase 4 trials are very much, and the parameters are very much collected. So here you will be able to see that for adverse drug reactions in the phase 1, it will be tested. And in the second phase, it will be tested for the efficacy and side effects. And also in phase 3 trial to confirm the efficacy and safety before it goes for the drug approval and marketing. So this is the steps that are very critically followed. So here I am very curious about the human phase 1, phase 2, phase 3 is generally if starting from 100 people, 100 or 50 healthy volunteers are going to test in phase 1. Similarly, 1,000 to 10,000 are going to test in the phase 2. And then in phase 3, all over the world in different parts, in different ethnicity and different geographical regions, the drug will be tested and the parameters will be collected. So here the genetic components are not going to be tested at all. It is across a common population. Healthy individuals will be there, disease expressed individuals also will be there. Against them it will be tested, and they are going to arrive at a final decision, and they are going to go for the drug approval. So the advances in the development, and today we are using so many computational techniques we are using for the analysis of the data, analysis of the genome data, identification of the mutations, and then very critically comparing what exactly is the change that is happening with the metabolic pathways. And thereby, with all this information, we will be able to very critically see, pinpointedly, we will be able to talk about which gene is involved in the disease expression. In other words, which protein is involved in the disease expression, one will be able to very critically scale down. So even the FDA has given guidelines regarding the precision medicine. So advances in precision medicine have already led to powerful new discoveries and FDA approved treatment that are tailored to specific characteristics of individuals, such as a person's genetic makeup or genetic profile of an individual tumor. So, for example, all of us are aware that whenever we are going to meet a doctor, if it is a simple cough and cold, or if it is a very simple seasonal problem, means then without any investigation, he's going to give an antibiotic or any other bronchodilator or any other, depending upon our cough and cold and other viral flu problem, we will be given a prescription without any pathological test. But on the other hand, if we go with a very serious complaint where what exactly is the problem with our internal organs is not known, means then immediately a blood test will be prescribed, a urine test will be prescribed, a CT will be advised, or a specifically a MRI will be advised sometimes, or a scan will be. So these are the radiology and the pathological investigations will be advised. But on the other hand, today we have arrived at a situation where the day is not far that even in our pathological prescription, one more prescription we are going to get, that is the genetic testing. So patients with a variety of cancers routinely undergo molecular testing as a part of the patient care. So specifically along with whatever the pathological tests that we are going to do, now in today's scenario, some of the high-end hospitals where the molecular analysis and testing facilities are available, they are going for a molecular testing as a part of the patient care, and thereby very critically they will be able to identify which gene is mutated in the patient body and what exactly is the change that is happening, and which gene is upregulated, which gene is downregulated, and where the variation and what is the reactions that are altering in order to bring about the disease at the molecular level, one will be able to identify. And based on that, a line of treatment, you're not developing any new line of treatment or new tablet or new drug is not being developed over here. We should not mistake. Whatever the 5 to 6 different line of treatment that is available, so which line of treatment is going to fit the patient? Instead of the trial and error method, we don't have to specifically say something the doctor prescribes, and then even after taking it for 1 week, there won't be any improvement. Then again, we are going to go, and then we are going to say that the problem is the same, it is not improved. Then again, he is going to change the prescription and is going to give. So such type of trial and error technique need not be used over here. So wherein very critically the mutations that has happened in the protein and what exactly is the modification in the metabolic pathway can be very critically identified, and the targeted treatment can be provided with minimizing the adverse drug effects. So here, sometimes if you are going to see the WHO statistics, people dying with the adverse drug effects are more than the actual disease succumbing. So that is why it is very crucial and important to save the patients from unnecessary adverse drug reactions. Thereby, they will be able to, in the first round itself, they will be able to given a proper line of treatment. So one size fits all approach to medicine that is based on broad population averages. So this traditional practice often misses its mark because each person's genetic makeup is slightly different from everyone else, often is very important way that affects the health. So specifically, the advent of precision medicine is moving us closer to more precise, predictable, and powerful healthcare that is customized for the individual patient. So here again, what exactly is the translational medicine? I'm going to talk over here, that you will be able to see here the target, the right patient population. So this is most important. The right type of treatment has to be specifically identified, and the right type of treatment has to be given, and limit and anticipate the toxicity, renal toxicity or liver toxicity. So that has to be very critically monitored, and it has to be identified. So specifically, one will be able to decide the dosage to the patient very accurately. The dosage has to be identified, and then the best, depending upon the genetic markup, efficacy, and all the other properties, the right type of best candidate drug has to be chosen. So this is more important. So for that, we will be able to utilize some of the biomarkers, some of the SNPs that are there. So by using the ClinVar database or the DB SNP database, we will be able to identify whether the SNP that is identified in the patient, whether it is a known SNP or it is a novel SNP, we will be able to identify. So what is the goal over here is effective, rapid, specific transit between the different developmental phases of specifically the disease has to be controlled over here. The line of drug that we are going to provide should be highly effective, and it should be able to give a very minimum side effects. In other words, adverse drug reactions. So improvement of success rate at each phase of development has to be seen. So specifically, when a patient group is specifically monitored with a line of treatment, so the improvement has to be seen at every level, and the patient should get a relief from his sufferings. So decision making, and specifically by the very critically, we can use the biomarkers over here in order to group the patients. Which group is going to, based on the biomarkers, which group is going to receive what type of treatment can be critically identified, and accordingly they can be classified. So here what is going to happen, choose the right type depending upon the group they are. So this leads to better efficacy and safety of the patient. This will ensure the best efficacy, and the unnecessarily the patient need not suffer from the adverse drug reactions. So this is the summary of what we are going to today, what we are going to call it as from bench side to bed. What is bench side is whatever the genomic research that we are doing in the lab, it should be effective, it should be practiced at the clinical level, and it should be beneficial to the patients, so that he did not unnecessarily suffer with the adverse drug reactions. He should be straight away from day one, he should be put on the right drug. So whatever the points that I have given over here, so that can be specifically ensured from day one, and the best efficiency and the safety for the patient at all levels of treatment or in his hospital stay has to be ensured. So critically, you will be able to see here in this direction, the Human Genome Project and whatever the post-human genome development that we are seeing across the world is working at. This is the website of homepage of the Human Genome Project that I am showing over here. So it is maintained by the NIH, National Institute of Health, the USA, and this is called as the National Human Genome Research Institute in NHGRI, and you can visit this particular page. You can see the website address www.genome.gov. So and then one more database that I would like to show you is most important database is the Genome Reference Consortium. So the Genome Reference Consortium is also a very useful site. From time to time, they are going to release the benchmark genome database, so that against that we will be able to compare the patient's genome sample, and then we will be able to identify the mutations that has occurred in the patient sample. So this is again now the current reference genome data that we are using for clinical practice is GRCH38 we are using. Even 35 is also effective, but over that the 35 is recently released. And for comparing the patient sample, 38 is being used. So GRC stands for the Genome Reference Consortium. Human, what is the build? So build is now the latest build is 38 version. So then we have one more database of say the where the variants are identified from different genomes. So this project, in other words, it is called as the 1000 Genome Project. So this is again a very prestigious project developed where they have very systematically cataloged the mutations, and also they have annotated the mutations to specific diseases. So this also can be referred. Then one more facility that is available is the ENCODE, the Encyclopedia of DNA Elements. So all this time, even in the class till even today, we are going to teach the students that the human genome is made up of nearly around 99 to 98% interon and only 2% exon. And we are going to call that interon region as a non-coding region, and the exon coding is only the protein coding region. But the post-human genomic era, it led to a school of thought. Some scientists said that the 98% of the human genome that is available, only in the exon need not be, say, variations in the exon alone is not going to matter. So they started analyzing the non-coding region, especially the transcription and the transcription control regions at the 5' and the 3' regions. When we are going to go and analyze, even mutations in these regions are going to very specifically affect the level of expression of the gene. So thereby, this particular database is addressing the Encyclopedia of DNA Elements, means specifically they are going to analyze the transcription sites and transcription regulation regions is analyzed and searched for any variations. If the variations are there in this region, how the upregulation and downregulation of the gene is going to happen. So this is again a very prestigious database where we are going to get a lot of information regarding the transcription regulatory regions. So then we have one more database called as the catalog of, say here, the somatic mutation in cancer. So this is again specifically, it is a beautiful, very huge database where all the somatic mutations that are observed and collected and annotated, and they are very specifically correlated with the type of cancer. So this is again for the researchers and students who are working in the area of cancer genetics, they will be able to refer. Then here we are also going to have another prestigious database is called as the International HapMap Project. So what is exactly is a HapMap is the elucidation of the entire human genome has made possible the very specific effort to develop haplotype map of human genome. So the haplotype map, in other words, in short, it is called as the HapMap. So specifically, it is a tool that allow researchers to find gene and genetic variations that affect health and disease. So you will be able to see at a time 6 nucleotides or 8 nucleotides or a single nucleotide. In particular combination, they are going to specifically influence the functionality of a gene or they are going to cause the variation in the gene. So the DNA sequence of what they specifically we are observing in today's scenario is the DNA sequence of any two people is 99.5% identical, but only this 0.5% variation that we are going to see over here is the cause of the disease, or person say susceptible for a disease. In other words, the disease risk. So the analysis of these SNPs or haplotype blocks is going to give a greater insight into identification of a disease susceptible person, or we will be able to correlate whether that haplotype, we will be able to use it as a marker for the identification of that particular disease. So again, you have a very prestigious database where only the single nucleotide polymorphism is annotated and very systematically documented, is called as the DBSNP. So DBSNP contains human single nucleotide variation either in the microsatellite and small scale insertions and deletions, along with the publications of the annotation and to what disease it is correlated, we will be able to see, and also the population frequency and the molecular consequence, genomic and RefSeq mapping information we will be able to get, and also the effect of clinical effect of the mutations also. This information also we are going to get. So over this off late, they have developed one more database called as the human gene mutation database. Here you are going to very critically get the gene symbol, and also what exactly is, whether it is a missense or splicing, or the variation is there in a regulatory region, or whether it is a small deletion or a huge deletion. All this information we are going to get from the human gene mutation database. So then almost from 50 years, we have been referring for any gene identification for a particular disease. We are going to refer to a database called as OMIM. It is, in other words, it is called as the online catalog of human gene and genetic disorders. So this is again a very powerful and very curated source that we will be able to refer. So then we have one more database called as the database of genomic variants. So these are OMIM is there even before the human genome sequencing. OMIM was available to us, but only the data and the details have increased. But rest of the databases like HafMap, ENCODE, 1000 Genome Database, they're all recent databases. That is a result of the post-human genomic era that we are going to see. Similarly, a database of genomic variant is available. So by disease or by the gene name or by the chromosomal loci also we will be able to give. So for example, we can say chromosome 8 and we want to identify a region from say 7 lakh to 8 lakh, means only that region we will be able to call and we will be able to identify what exactly is the variation is there. Now again they have hooked in a new parameter, that is the environmental parameter, into the expression of the genome and diseases. So again a new line of database has come into reference, that is called as the Environmental Genome Initiative. So these are the remarkable evidences that we are having here. And now you know that the epigenetics, say 10 years back, if we were to talk about epigenetics, people would have thought us to be mad. What is she talking? But in today's scenario, with the type of analysis facilities that we are getting, the epigenetic factors are very critically controlling the expression of a gene. sometimes in our research only we have seen the coding region is not going to have, the exon region is not going to have any variation at all. Not even a SNP will be there. It will be a perfect gene that is expressing into a perfect protein. But still, the specifically a disease will be there associated with to that particular gene, or to that particular gene coding with the protein. So how do you explain this? Sometimes there are epigenetic factors that are going to very critically control certain genes and its expression. So there is a dedicated database for the human epigenetic epigenome project. In other words, it is called as the HEP. So wherein we are going to get specific epigenetic information for each and every gene that is available. So what I mean over here, why did I show all nearly more than a dozen databases, the recent databases that are formed after the human genome sequence was made available. So why the entire paradigm shift in research has happened, and identification of the variations at different levels, addressing at the exome level, addressing at the interon level, addressing at the epigenetic level. The reason is genomics has and continues to transform our world in myriad ways. So for example, any living organism on this particular planet, if it has to survive, whatever phenotypical characters or metabolic properties it is going to have, it is because of the blueprint that is there in the nucleus, that is the genome. So here, a genome is specifically a DNA present in the nucleus is not a mobile entity, it is a dynamic entity, wherein specifically the warriors or the soldiers of our body is specifically the proteins. So we have to translate the genomic data into a structural genomic data or a functional genomic data. So that can be done only by the DNA sequencing. So a lot of development with respect to equipments and protocols have happened. The Illumina has, every 6 months, Illumina is coming out with a advanced version of the DNA sequencing equipment. So advances in genomic are reducing the cost of genome sequencing by millionfold. So where is $3 billion and where is 25,000? So human genomic variation, identification of the human genomics mutations or variations are helping us to understand what makes us different from and what makes us the same. And rare genetic diseases, the cause of the rare genetic diseases and the diagnostic odysseys for the patients with the rare diseases can be formulated. And especially the cancer, because of the development of special cancer databases and cataloging of the cancer causing genes and mutations, the line of cancer treatment itself has taken a complete reviewing and reviving. It has happened. New drugs are available in the market where very specifically highly target oriented, and only the cancer cells will be blocked and not the healthy cells. So thereby genomics is transforming the study, diagnosis and the treatment of cancer. And when it comes to pharmacogenomics, genomics is helping us to choose the right medication at the right dose for each patient. And we are also going to see certain non-invasive prenatal genetic testing. So the knowledge of genomics has revolutionized. So at the say a pregnant lady without causing any invasive collection of the sample, in a non-invasive way, one will be able to do the genetic testing, and this is revolutionizing the health assessment of the newborn, the fetus, and also in ensuring a healthy child delivery. So now, all of you are hearing about the CRISPR genome editing. Genome editing is altering the genome with unparalleled efficiency and precision. We will be able to edit with the CRISPR technology. So this is also very specifically the enhanced forensics. It is also applied to other fields and microbes and microbiome, sequencing of the genome of the microbes and microbiome. Today, if at all, if we are safe, even after the COVID, 2 to 3 rounds of COVID attack on the suffering of the people with the COVID pandemic, and also within a short time, if we are able to get the viruses because of the technology that is available in India, wherein within a few days itself, within 1 week, we could know the genome of the COVID virus and also the proteins that are going to be formed over there. Keeping that, with the constant effort, the scientists developed the vaccine. So today the genomic testing is directly from our home, only by our courier we will be able to send the sample. So that is called as the direct to consumer genomic testing facilities are available. We don't have to go for any pathology lab door, or even if a genomic testing facility is not available in the city where we are in, we will be able to send it by post. So the genomics help us to understand evolution and order also, not just in the biomedical field alone. Genome is not of importance. Even if the environment is safe, we are safe. If the environment is healthy, we will be healthy. So in order to see that the environment is healthy, so genomic mapping and analysis of the evolution also has to be done for the biological ecosystem. So the various facilities are available over here. This I have given it as a collage over here. The ClinVar, CPIC genes, pharmacokinetics, genotyping, and then specifically the different facilities that are available for the precision medicine. So the genetic variations like SNPs, CNVs play a key role in drug metabolizing enzyme. So when once a drug enters our body, how it is metabolized is very crucial. So every individual is going to metabolize the drug in a different way. We cannot normalize the drug metabolism. So the drug metabolism is going to play a crucial role in expressing an adverse reaction or a recovery or any type of toxicity in the body. So this is a very crucial point that has to be very critically studied. So the SNPs and the CNVs are going to play a crucial role. In other words, the protein that is going to be expressed by the gene with these variations are going to play a very crucial role. So DNA sequence has a major impact on the efficiency and toxicity of the treatment. So here the base pair level of a genomic is specifically 99 some of the papers they are going to give 99.9. So we will take it as 99.5%, the same for all humans. And this 0.1% difference are many of the things to make each and every individual unique. So genomes differ from each other in all sorts of ways. So those differences in DNA help to determine phenotype and risk might be for various diseases. So for example, some of them may be susceptible for certain disease in a family, but on the other hand, some of them will not get any disease. And also if we are going to see the ethnicity, for example, say in the American population, whatever the Jiva studies that I have made, Caucasians are susceptible for certain type of disease. American Africans are susceptible for another type of disease. And similarly, the pure Americans, again, they are susceptible for certain type of cardiovascular diseases, obesity and diabetes. So thereby the ethnicity of a specific population is also going to play a very critical role. So here, what exactly is the translational medicine is whatever we are going to talk at the bench level, that is whatever we are going to do in the research laboratories. We are going to talk about the genome, mutation, and whatever disease we are going to advocate because of the mutation. So that information has to reach the clinical practice, and the doctor has to consider those SNPs or copy number variations that is there in clinical practice. It has to be used and a tailor-made line of treatment, and it should reduce the burden of suffering of the patient from the adverse drug reactions or side effects. From day one itself, if he is put on a particular line of treatment, that line of treatment should be beneficial to him, and that should not cause any type of adverse drug reaction in the patient. So this is the message, and this is what it has to be practiced. In other words, it is called as the translational medicine. Whatever research that is done in the research laboratories, there has to be a continuous interaction between research and clinical developmental activities, and thereby the whatever the information that is discovered here, it has to reach in the patient treatment. So previously, all cancers were treated by a single line of treatment, but today doctors are trying to identify very early in the before starting a line of treatment. They are going to very critically identify to which patient subpopulation the newly enrolled patient is going to belong, and thereby he can be very critically put on a line of treatment that he should be able to respond effectively from day one itself. So this is called as personalized medicine. Today we have targeted therapies for some cancers, especially the immunotherapy that is being given to the cancer patient. So this is completely a personalized line of treatment with response rate is nearly 90% cure or control of the disease we are going to see with respect to the cancer patients. So I'm going to show you certain case studies that we did in our lab. Nearly the mapping and analysis of SNP in exon and UTR regions from whole exome sequencing data for diabetic patients. This project we undertook. So what did we do is in a very simple way we did it. This was funded by the DBT. So nearly around 12 patients we selected who are well diagnosed with kidney problem and who are all chronic diabetic patients. So we took nearly around 5 ml of blood from each patient, and then we extracted the DNA and using the Illumina HiSeq platform, we sequenced the entire genome of that individual, and we analyzed the genome data for the SNPs. And this is how the protocol that we followed I have given over here. So annotate the variant calling files, and then the CSV files were generated. And finally, the genes that are having critical mutation, that are showing mutation, were identified. So for example, if we are going to nearly around 90 genes were identified in the first round. So type of mutations in the 12 subjects, we could scale down to 90 genes over here. So non-synonymous, 58 genes, they showed a non-synonymous mutation. 14 genes, they showed the frame shift. 37 genes showed the non-frame shift mutation. And 3 genes showed the stop gain mutation. and we could not annotate 29 genes with unknown mutations, but serious changes were there. So with this, I'm also giving you the list of genes over here on the in the first column. This is the serial number. Whatever LMC you are seeing over here, this is the patient ID. So this is the patient ID that we have maintained. And we have done a classification over here that how many patients where two or three patients are showing the same type of mutation for one particular gene, and nearly all 5 of them are showing same type of mutation for another type of gene mutation variation. So this is how we classified the data, and this is the raw data that I'm showing over here. So which is the gene and what type of mutation has happened to him. So this is automatically by the latest whole genome analysis software, we are able to generate this, and I'm showing this over here. And this is again a further analysis where what type of change has happened, whether it is a domain shift is there, or a motif shift is there, or a motif is modified, or a secondary structure of a protein is modified. So here you will be able to see one sample, the gene name is MEATN3. You can see here the domain is modified. Loss of calcium binding domain has happened because of the mutation. And you can see the what was in the normal gene, a turn was there, and in the mutant, a long helix is being introduced. So in one more gene, you can see over here PDE11A. So the frame shift mutation has occurred over here, and the all functional domains of that particular protein is deleted because of the mutation. And this is a very serious change that we are going to see. This is how nearly all the 90 genes we analyzed. And when we constructed a pathway over here of the 90 genes, we could very critically see that almost all the 90 genes are very specifically involved in the signaling pathway of insulin. So you will be able to see over here, this is the insulin gene that it is there in the center. And all these genes that are identified with mutation in the patient. So when a enrichment map was developed, we could very clearly see that all the genes are, yes, with the mutation. They are very much in, say, in the pathway with the insulin, but they are there in the signaling pathway of the insulin. So this is how we will be able to further analysis when we did is we gave a differential coloration where the domain loss is there, and a change in the domain, deletion with the domain, and change in the functional sites. So we are going to see a lot of modification has happened with the protein functionality. So at the grassroots level, when you're going to take the PKD 1L3 gene, you will be able to see over here the specifically the functional motifs are going to change over here. This is the normal one, and this is the mutated one that you are going to see over here. And you can see the length of the it has changed over here. And very critically, the shift in the functional motifs. See here, how many motifs are there in the normal, and in the mutant you will be able to see nearly around 4 to 5 functional motifs are completely decided. You concentrate here. 1, 2, 3, 4, 5, 6 motifs are there over here, whereas in the mutant one we are seeing only the 3 motifs are there, rest of it is completely deleted. And when we go for the protein 3D structure development, this is the normal protein. See, look at the size of the protein over here. And because of the mutation, the protein structure is this. So it may be synthesized, or it can be stable, or it may not be stable. That we have not verified over here. But there is a very drastic change in the protein 3D structure that we will be able to see. This is another protein. So wherein very critically we will be able to see, this is the normal protein that we are seeing. See the functional motifs that you will be able to see here, and in the mutant very critically we are going to see a change. And in the 3D structure also, we are going to see a very definite variation in the 3D structure. So then in some of the patients, we could identify the recessive homozygous haplotype block that is very critically related with the genes involved in diabetes. So you will be able to see here the haplotype blocks that is marked over here. Very critically, the mutations were identified over here. So the type 2 diabetes patient sample was taken, and 20 to 40% of the patient population, they had the diabetic nephropathy problem. And this was completely ensured by the clinical diagnosis regarding their disease. And when the variation studies was done with respect to the SNP, structural variation, copy number variation, haplotype blocks, and then the mitochondrial variation. So we could see that all of them, they showed a very remarkable haplotype variation. You can see over here in, see this, whatever I have marked in rectangle over here, you will be able to see that the patient sample had a very critical haplotype region, and he is also, the patient is also a chronic diabetic patient, plus also having a serious kidney problem. So what is the gene that is showing a haplotype block is chromosome 5. Nearly the homozygous region count, you will be able to see 10 over here. A number of genes identified is 17. So this is a very serious mutation that is being observed here. So where the gene is located, specifically in that particular region, the PCB D2 gene is located. And in chromosome 6 also, some of the haplotype blocks were identified in the NEU, SLC4. So this is how we could very critically identify the haplotype over here. This is another analysis. So in this region, you are going to see here a haplotype block, wherein nearly, it's not a small chunk of nucleotides, it is nearly roughly around 5,000 base pair of nucleotides that form the haplotype block over here. So this is how, these are some of the analysis that is done on the other genes associated with the diabetes over here. And clearly you will be able to see the number of genes that are associated with the diabetic disease also can be cross-verified over here. So when using the Ingenuity pathway analysis software, when we were able to plot a pathway map, so this is how we will be able to plot the map, and we can very critically understand by the pathway map that all the genes that has undergone mutation are critically involved in the genome metabolism, in the insulin metabolism. So genomic information, susceptibility of an individual to certain diseases, so that adequate steps can be taken to minimize the risk associated with these diseases. So here we are very critically in the TCF7 L12 gene, we are seeing a significant mutation and higher risk of developing type 2 diabetes, it will indicate. So today, most of the genomic sequencing companies, for some of the common diseases, they are having gene panel. That is by research, certain genes and the mutations, 100% they are identified and linked to the disease formation. So based on that, the patient sample need not be compared with any of the general domain data. Against this gene panel, one will be able to run and one will be able to identify where is the mutation in the patient body. So what is going to happen? Let me come back to my original discussion that I was doing with the personalized medicine. So knowing the specific genes that has undergone mutation, we will be able to take the preventive and definitive therapeutic approaches. So they will be able to take care. For example, say in preventive medicine, we are going to say that if at an early age, if we come to know that we are having a mutation in a particular gene means, then we will be able to say design or care can be taken to have a specific type of lifestyle and to avoid certain environmental conditions. So all this care can be taken so that the gene expression is not triggered over there. So then minimizes the line of treatment. When we are going to come to the line of treatment, by knowing the mutation in a particular gene, a right type of treatment can be given that is going to minimize the drug adverse reaction, and then allows dosage and dosing frequency adjustments can be done. So for how many and how many hours gap a particular drug has to be given, and the what quantity should be given. So dose and dosage adjustment can be very critically decided upon knowing the type of change that has happened. To conclude with, what is the summary of my talk today is, personalized medicine has resulted in a paradigm shift in many drug development programs by allowing the deliberate enrollment of patients who will benefit most from various unnecessary experimental interventions. To try one line, it's not working, then you advise one more line of drug. That is not working, advise the third line of drug. So it is going to save the patient from unnecessary trauma. So the concept of personalized medicine have existed in healthcare industry for a very long time. But however, due to the technological advances and improved data processing in the last few decades after the Human Genome Project, now a deeper understanding and importance of pharmacogenomics is identified by the industry, and which has helped the developers and physicians to implement personalized medicine concepts in clinical practice and also in real world treatment. So additionally, advances in DNA sequencing with respect to speed and cost. One more perspective is the technology should be user friendly, the technology should be accurate, and at the same time, it should be easily affordable by especially the medical treatment itself is so costly. Everybody will not be able to afford the medical treatment for a long period of time because of the financial constraints. But when a particular line of investigation, it becomes cost effective and cheaper, then only it can reach the common man. And also within a short time, the result should come. So you are going to give the blood sample, and after 1 year or 2 years, if the result of that sequencing is going to come, means it is of no use. Instantaneously, within 48 hours, the information, analysis information should be available to the patient. And the doctor also should be able to decipher what is given by the genetic lab, and thereby he should use that information for better diagnosis and treatment. That is the bottom line of my lecture that I am talking today. With this, I conclude. Thank you.
Ma'am, I have a question for you. Personalized medicines are very expensive when compared to traditional medicines because it involves companion diagnostics as well as genetic testing. Then how do you overcome the cost issue, like the cost challenge? Because the poor cannot afford.
Yeah, I fully agree with you. I fully agree with you. Even in Bangalore today, we are giving the samples for somewhere around, say, plus or minus 30,000 rupees we are giving. So it is not affordable by each and every one. The cost of genomic sequencing still has to come down. But in place of going for genomic sequencing, today one more facility that is available is the target sequencing. A disease, if we very specifically know, for example, breast cancer means BRCA1 or any other line of genes will be directly, it will be sequenced. So such type of facilities and that's called as the targeted sequencing, that can be done for a specific disease. So that is going to cost somewhere around 3 to 4 or less than 5,000. So that is highly affordable instead of going for the whole genomic sequencing. Yes, medical treatment in our country is very high. Unfortunately, education and medical treatment should be free for everyone. Then only a country will prosper and a country will be healthy. Educated population, healthy population are the two pillars of a country. When a society has to be productive, means it has to be healthy and it has to be knowledgeable and skilled. But unfortunately, the education field and the health field are the two most neglected fields in our country.
Thank you, ma'am.
Hello. Ma'am?
Yeah, I'm hearing. Tell me.
Yeah, ma'am, you were speaking about this diabetic nephropathy and NGS, that whole genome sequencing. So, is that one published, ma'am, that that has been published?
No, that we have done only with respect to 12 patients.
Okay, ma'am.
That validation has to be done. For a larger
Validation of
population,
this.
yeah, on a
okay,
larger population, we have to do it. That we took it
Okay.
as a pilot project for a
Okay.
diploma program. We had we
Okay.
have a diploma program in our department called as personalized genomic medicine.
Okay.
So in that course, we took this pilot project and we did it. But if we have to publish a paper, we must have quite a number of patient sample has to be analyzed.
Yeah. So you will be analyzing only for those
No,
novel
we are
mutations which-
proposing, we have to validate these results and then we have to
Okay.
publish. We are waiting for it.
Okay. Thank you. Thank you.
Madam. Hello, madam.
Yeah, yeah.
Madam, I'm asking you whether the success rate, how much is the compared with the other medicines the persons are taking? How much is the success rate and the lifespan?
No,
and the
especially
lifespan?
with respect to cancer, you will be able to see the classification of the patients are done. And today, you are hearing some of the celebrities, they were diagnosed with cancer and still they have survived and totally cured, right? So at the same time, initial identification of the disease has to happen. From day one onwards, they have to put on the right line of treatment. Success rate is quite high with respect to precision medicine, especially with
So, at
respect
the first
to,
level, we have to start the medicine. Isn't it?
yeah,
We have to start the medicine. Thank you, madam.
okay.